This episode is dedicated to the memory of Jay Nichols. Jay was a true mover and shaker in ocean conservation and the study and protection of sea turtles. His Blue Mind concept and initiative has inspired all of us at the CheloniaCast to embrace water and its healing power. Jay will be greatly missed. This is CheloniaCast, episode 67. Today we are joined by Dr. Jeff Seminoff, who's a program lead of the Marine Turtle Ecology and Assessment Program and director of the Stable Isotope Laboratory based out of the NOAA's Southwest Fisheries Science Center. Dr. Seminoff has been involved with sea turtle research for over 30 years. He's got a really impressive resume working with sea turtles in all capacities. He served as the past president of the International Sea Turtle Society. He's a current U.S. delegate for the Scientific Committee of the Inter-American Convention for the Protection and Conservation of Sea Turtles. He's also the executive editor of Colonian Conservation and Biology and multiple other journals, which is a really cool crossover. You don't typically see folks working with sea turtles in sort of the freshwater turtle and tortoise realm. So that's really an interesting area. We'll sort of expand upon later. He's the author of over 200 research papers and the editor of the book Sea Turtles of the Eastern Pacific. We're really excited to talk to him today about some of his work with sea turtle conservation and research. So thanks for joining us today, Dr. Seminoff.
Michael, thank you. It's a pleasure to be here.
Awesome. Well, I'll just kick it off. The first question we like to ask everyone, I think it kind of, everyone's got a different story, but how did you end up working with turtles, and have you always been predominantly interested in sea turtles? I mean, where did the journey begin?
Yeah, good origin question. I've been a herper, turtle, reptile, amphibian lover my entire life since I can remember. Even to this day, my wife gives me guff because anytime I see a critter, I try and catch it. Just that's in my DNA. I try and catch animals. But as a young kid, I lived in Illinois, caught painted turtles, had extended family in Northern California. So I'd go there every summer and focus on like catching lizards and snakes and stuff like that. And I was one of the fortunate ones that I knew that I liked biology as a high school student, as well, really before high school. And so when I went to college, and I went to high school in Connecticut in the northeastern United States, and went to college at the University of Arizona, which is pretty much across the country, about almost as far as you can get from where I went to high school. And I remember distinctly going to college, and I learned this new term that I'd never heard before, and the term was ecology. I'm like, wow, ecology, like the study of animals and plants and their interactions. And it just intrigued me from the first time I heard it, and it was like, sign me up. And I was a biology major. I quickly changed my degree to ecology and evolutionary biology. And in those early years, I had worked with a lot of different species, kind of even as an undergraduate, track and desert tortoises, working with spotted owls, working with chuckwallas for the Arizona Sonora Desert Museum in Tucson. And I had an opportunity to travel to Central America, to Costa Rica in 1986 to work with green turtles at a place called Tortuguero, which is started by Archie Carr in 1954. It's one, if not the longest running sea turtle conservation programs in the world. And that was sort of my first dive into sea turtles. And I just loved the taxon. Little did I know I'd be studying turtles later on. But then I ended up, I stayed at University of Arizona. I did sort of an atypical thing. I stayed there for 3 degrees. Do not try this at home, everybody. But for mitigating circumstances, I did my undergraduate at U of A, took some time off, started an environmental education nonprofit called the Coastal Conservation Foundation, and ended up staying in Tucson and worked on my master's and PhD there. But during my master's degree, I had actually was going to study reef fish recruitment, so marine biology of fishes in the Gulf of California in Mexico, which is also known as the Sea of Cortez, which is the closest body of water to Tucson, Arizona. And it's a little bit of a long story, but it's sort of my origin story, so bear with me. But at the time, I met a colleague named Jay Nichols, who was a recent undergraduate from his undergraduate degree at Duke, and we both made fast friends. And I said, hey, Jay, let's go down to Mexico. Let's check out some nesting beach. Let's go dive in Mexico, because he was going to study moray eels. And we're on the beach on a place called Venecia one evening. This is 1992-ish. And we both just learning about each other. We mutually discovered that each had worked at Tortuguero in Costa Rica. And I was there in 86 and Jay was there in 1990. And drinking beer, drinking tequila on the beach. And we said, wouldn't it be cool to go visit like a nesting beach in Mexico? Because here we are in the US bordering Mexico. And at the time we're in Mexico having our libations. And that quickly, after a couple more shots of tequila, transferred into, wouldn't it be cool to visit all the beaches of Mexico, which is a preposterous idea. But finished up the night, did a bunch of diving the next few days, went back to Tucson for me to start work on my master's degree on reef fish and Jay on moray eels. And we rekindled that conversation and said, hey, wouldn't it be cool to maybe do that and go check out some beaches? And little by little, to abbreviate the story a bit, after about a month and a half or so of us investing so much time on planning on this turtle trip, that my major professor at the time, a foremost ichthyologist named Don Thompson, said to me, you know what, Seminoff, you're spending so much darn time focused on this turtle project. Why don't you drop fish and just study sea turtles for your master's? And that was all I needed, basically. And so that's the initial sort of dive into turtles for that project. We focused on nesting beach conservation strategies and the effectiveness of those strategies, whether or not they worked with local communities, and just the dynamics of conservation biology at sea turtle nesting beaches. And throughout that process, met a lot of different people. And towards the end of the trip, we stopped in a small fishing community, the former sea turtle fishery capital of Mexico, called Bahia de Los Angeles in the Baja California Peninsula, and was welcomed with open arms by the local sea turtle scientist there, Antonio Resendez, who became a mentor and father-like figure for me. And after my master's, I didn't want to waste time going to another school, so I just transferred into the PhD program at U of A and started studying not nesting beach ecology, but in water, or studying sea turtles at sea, which at the time, 30 years ago, was really unusual. Most people studied turtles on nesting beaches. And so that's kind of initially how I got into sea turtles, starting in 1993, and then the in-water work starting in 1995. Lots of crossroads since then, but here I am today, looking back on a life that I'd never trade for anything else. I wouldn't do anything different.
That's an awesome summary. I think maybe we could just jump into some of the interesting, what it was like to conduct your thesis work and such in the Gulf of California. And you mentioned that there wasn't a lot of in-water research prior to your 1995 work there. And in that region, I can't imagine there was a lot of information baseline on the population of green turtles there. And I'm just curious, your thesis is very holistic, ecological, and you look at the dietary analysis, the movement ecology of the sea turtles there, and there were some interesting patterns. I'm curious, though, what the most interesting takeaways from that were, and what you really learned in the process of doing that work.
Sure. So rewinding to 1993, 1994, this was like the birth of the internet and email, and I honestly don't know if I even had an email address at that time. And so information was really hard to come by. It's what you could get from voice or from colleagues or the hard literature, hard copies of things. And I knew that Baja was an important place for turtle ecology, but also sort of one of the epicenters of a sort of a conservation disaster that was happening through the 60s through the 1990s. And that was the extraction of green turtles at an industrial scale. With the town that we worked in, Los Angeles Bay, so I started there in 1993 was the first time I was there. And it was only 3 years prior, in 1990, that the president of Mexico, President Salinas de Gortari, created a presidential moratorium on any use of sea turtle products, both for medicinal or for food uses. And so his decree was actually probably a decade too late. The populations of turtles had absolutely crashed by that time. Still, there were turtles. And to give an example, just the culture of what it was like back then, in that sleepy town of Los Angeles Bay, you could walk down the street on any given day, 365 days a year, and you would smell that very familiar odor of cooking turtle. This was a town that didn't have roads until the 1940s, 1950s. It had human occupation since the gold mining days in the late 1700s. And so they used the local resources. And one of them, of course, is turtles. And as we all know, sea turtles are, from the Columbus days, are a great food stuff in that you can capture that animal and keep it in a cool, dark place, and they'll stay alive for months. And so you can have the stockpiled turtles that can provide a food resource for a very long time. And Los Angeles Bay was no different. Like, turtles were the primary protein staple outside of fish. And I sort of, it's not a joke, it's a true metaphor that back then, if I was to go into that community and say, stop eating turtle, it would be no different than going to Main Street USA and telling everybody, you cannot eat hamburgers anymore. Same exact thing. Like, it was such a, like, the conservation ethic was very different. This idea of exhaustible resources was not prevalent yet. So it was still this like inexhaustible food stuff that people could get as much as they wanted. And so for me, my biggest lesson was not necessarily the biology, but it was like working with people and understanding other people's perspectives and putting myself in their shoes before I pass judgment. And for me, that sort of sensibility helped me really, I think, make inroads with the community. Because at the end of the day, for all the work that Jay and I, and largely me in Los Angeles Bay, the work that we did, it really relied on locals. So I had several different informants that were former turtle fishers at the day of the legal harvest, which is 3 years prior, as I mentioned. They taught me how to build turtle nets, how to mend turtle nets. Again, this is for capturing turtles in the water. They taught me how to read the tides, where to set nets, how to get turtles out of nets. Everything that I know today, really, the foundation is those fishers in those local communities that weren't scientists, but they were as good sea turtle natural historians as anybody else because they lived it every day. And so that aspect of just that cultural sensitivity, I think, to me, that's the biggest take-home message. And that's what I try and convey to students and colleagues as they're working in places that are not in their own backyard, you know, as much as possible. From a biological standpoint, I think that to me, the most interesting thing with turtles in LA Bay and elsewhere, I think it really sums up to one word, and it's resiliency. And so after literal decades, if not a century of harvest throughout northwestern Mexico and in Los Angeles Bay, for example, from the 1960s through the late 1970s, roughly 500 sea turtles were shipped out of the community every 2 to 3 weeks. So tens of thousands of turtles taken from that area. Yet still there were turtles in that area, and I was able to do research and get my work done. Now, granted, the populations were much lower than they are today, or much lower than their former levels, so it took me a while to get sufficient sample sizes, but still there were turtles there doing their thing. And so to survive that onslaught, to me, is sort of a lesson that I take home, even in my own work. That resiliency is something that I think is really important. We can all learn from turtles in that respect.
Right. Yeah. I want to go back to the cultural aspect because that's really interesting. And I think I listened to there was a recording with Dr. Nichols from a while past, and he talks about the Seri tribe, and one of the tribes that's sort of the cultures in northwestern Mexico, and kind of the esteem that they hold turtles with there, and what it means, a single adult turtle and the eggs and that sort of thing to the community. I mean, they may be eating it, using it for food in certain capacities, but it seems like a lot of the cultures in northwestern Mexico, it's sort of a sustainable practice. And I mean, what are some of is that true? And what are some of the specifics you took away from how they sort of look at turtles there?
Yeah, so the Seri, I think the contemporary term that we use is the Comcac tribe. So that's
Okay.
the non-Anglo term. That's what they call themselves. It's a tribe in northwestern Mexico that present day occupy the state of Sonora, Mexico. Earlier in my career, I had the opportunity to work very closely with that community. Well, there's actually two communities, Puntachuec and El Desemboque. The Comcac are a seafaring people. They're built on the protein of turtles, to be honest. If you were to visit the Comcac community today, you would see some, in terms of their stature and their physical form, a little bit atypical of what I think we often think of when we think of indigenous tribes. They're very tall, they're very slender, they're very powerful and muscular. The women are 6 feet tall. Very large people. There are some thoughts that that sort of is the product of millennia of consuming high protein diets, that it sort of promotes like larger body size. I don't know if that's true or not, but that has been explained to me by some ethnozoologists. But Sarah, I think it's a good example. I used the fishers of Bahia as an example to talk about how fishers can be natural historians and know as much as the scientists. And I think the case with the combcock, like other seafaring turtle using indigenous cultures, is they're better biologists than the Western scientists are. And so for the combcock, for example, they identified 9 different species or 9 different variants of green turtles, 2 different forms of loggerheads, 2 different forms of hawksbills, leatherbacks, olive ridleys, and loggerheads. And so those 5 species occurred in the region. And they had a deep knowledge about the biology and the seasonality of those animals. The green turtles, Musni as it was called, that was a species that was harvested using a spear, sort of a harpoon sort of a format for food. They would also catch leatherback sea turtles. And leatherback sea turtles, actually, that's sort of from the combcock origin story, is that they believe that their ancestors are leatherbacks, and that actually their whole culture is based on sort of a lineage that started with leatherbacks. And so when, unlike green turtles that are consumed, the combcock with leatherbacks, well, I'll get to this in a second, but when they would catch them, quote unquote, they'd bring them to shore, an individual. They wouldn't kill it. They'd bring it up on the beach. They would adorn it with natural pigments, paints, put it in shade, and they'd have a 4-day ceremony. Leatherbacks are called musnipo in the combcock idiom. And after those 4 days, those animals would be released. Now, if you were to talk to the elders, and some of this has been conveyed to me by my late mentor, Richard Felger, who's probably the foremost ethnozoologist of the combcock community. But as the elders in the combcock tribe would say, is they actually didn't have to catch the leatherbacks, that they would sing and the leatherbacks would follow them to the shore. And the leatherbacks would actually kind of crawl up on the beach, and then the combcock tribe, the members would pull the turtle the rest of the way and then build the shade hut. So in 1999, I, along with Scott Eckert and a guy named Hugh Govan, held a workshop in the town of El Desemboque, where we were talking with the combcock elders and trying to figure out kind of where was that intersection of combcock knowledge with Western science. And at the end of this 3-day meeting, they said they wanted to take us out to a spot in the Canal de Infernio, the Hell's Channel, which is like sort of the area right adjacent to their homeland. And they wanted to take us to these areas where turtles live, these turtle homes. Musne Ime is what they were called. And it was a really snotty day out, a lot of wind, rain, clouds, not a day that anybody would want to be on the water. And the typical boats that we use are these 22 to 24 foot open skiffs with an outboard motor. Anyway, we had 3 boats. There was probably 15 people to 20 people, some close colleagues of mine that today are very active in the conservation community in Mexico. So I have witnesses to the story I'm about to tell. The story has also been cited in Carl Safina's book on leatherback turtles. And it's kind of when anybody asks me, like, have you ever seen anything that is unexplainable that you have no idea how to happen? This is the story. So what the story is we went offshore to the Musne Ime. It was completely rough outside, not going to see anything. And turtles, green turtles in the Gulf of California, are extremely skittish. They don't like to be around people. They never have been. Probably that's a function of like unnatural selection. Like the ones that survived were the ones that were really skittish of people. But we went out there to the spot, and we tied bow to stern, bow to stern, bow to stern to have like a triangle of boats, a little flotilla right over the Musne Ime. Really windy and couldn't see the sun at all. And the 2 elders, one gentleman named Gabriel Blanco, who was the famous turtle hunter. I have images of him we can share if we want to. And awesome. one Jose Luis, who was the eldest combcock member. They started to chant. And I've told the story many times, and the hair on the back of my neck stands up. But they started to chant. The winds calmed. The skies opened up above us. The sun shone through upon us. This is not a joke, folks. This has literally happened. There's witnesses. The sun shone through, the seas calmed down, and green turtles starting popping up around the boat, kind of bobbing up and down on the water. There's probably 5 or 6 animals completely transfixed on our pongas and on Jose Luis and Guadalupe's chanting. And they just sat there and just were not afraid of people at all. And we are just dumbfounded on the boat, could not believe this was the case. Went on for what seemed like 5 to 10 minutes. I really couldn't tell you how long it was because we were all just so amazed by it. But then Jose Luis and Guadalupe stopped doing their chants. The turtles kind of didn't turn around and swim away fast. They just kind of seeped back into the sea. The skies closed up. We lost the sun. The wind picked up and the water picked up. And it's like, did that really just happen? But it did happen. And so when you ask me, did the Comcock sing leatherback turtles? Did Comcock sing the Moose Neepole to shore for these 4-day festivals? I can't tell you that they didn't. It's very possible they did. And that's that fifth dimension or whatever that, these things that we just might not have the senses to pick up. So
that's fascinating, right? And yeah, as you said, the local knowledge is so broad, and you have to think that a lot of sort of Western science has only been in these regions for a few hundred years, whereas there's so much more rich history and long-term observation that's there to, to kind of contribute to the overall understanding of an animal. Someone that Robert Villa, it's someone that's he works with a lot of herpetology, general Sonoran herpetology, and I've talked to him a good deal about just all the different varieties that different sort of cultures recognize in the north of Mexico and the desert, and it's just really fascinating and great collaboration. There's a lot there to be learned. Something else that's really interesting, kind of related to the Gulf of California, that Baja California in general, is that there are loggerhead turtles there, and that's something maybe that isn't. I think we think about East Pacific as very heavily associated with green turtles, but there are loggerheads as well and other species. And early on, through your tagging efforts and that sort of thing, that there was a huge discovery that there was a loggerhead that had literally migrated from the Gulf of California all the way to the coast of Japan. And that's a pretty incredible migration. What was it like to find out that someone had found the turtle that you tagged across the Pacific?
Mm-hmm. Yeah, the turtle you're speaking of, its name was Adelita. It was a she. We know that it was actually caught in a fisher's net just adjacent to a sea turtle nesting beach after it had finished the 10,000 kilometer trans-Pacific migration. This is back in 2007-ish. My years might be a little bit off there. But so to back up, loggerheads have been seen not in great abundance, but off the Pacific coast of Mexico for many, many years. There's no known nesting beaches for loggerheads in the Eastern Pacific, whether it's the coast of North America, the coast of South America. And for the longest time, scientists simply thought that, well, loggerheads are here, they must nest nearby, and we just haven't discovered the nesting beach yet. And then in the late 1980s, a turtle bearing a flipper tag from Japan was encountered on the Baja California Peninsula, a loggerhead. This is, I think, actually the first paper that Jay Nichols and I ever wrote together was the recount of that one tag. But we realized that, wow, they might not be nesting in the Eastern Pacific. In fact, they might be coming all the way from Japan. And right around that time, 1992, 1993, a foremost geneticist, Brian Bowen, and his colleague Alberto Abreu, American and Mexican respectively, did genetic work, and they did indeed establish that loggerheads that are found off the Pacific coast of Mexico share the same mitochondrial DNA haplotypes with the loggerheads that nest in Japan. Interestingly, there's an analog in the southern hemisphere in that there's no nesting off of Ecuador, Chile, Peru, but there's loggerheads in that region. And through genetic work and telemetry work, we've identified that those turtles actually originate from Australia and New Caledonia. And they do this crazy trans-Pacific migration down south as well. And so for us, that with Adelita, at least, that's the first sort of tracked example, of a satellite tracked example of a trans-Pacific migration of loggerheads. And say what you want, like flipper tagging, I think it's a pretty unequivocal way to establish connectivity. The tag gets placed on a turtle in one spot, it gets recovered, the turtle at another spot, and you can connect the dots A to B. But there's something about a satellite track, something about that human connection with actually watching the animal's movements to be able to see it sometimes in real time, daily as they're making their journeys from these long distance journeys from maybe it's from the foraging area to the nesting beach, as in the case of Adelita, and that's something that I think is of all the science dissemination that happens in the turtle world, I will say, in my belief, satellite tracking maps are probably the thing that lay people most identify with, because you can just get it. You can see the map and you can see the tracks. And so in the case of Adelita doing that trans-Pacific migration, this came out in a Bulletin of Marine Science paper in 2000 that Jay first authored. It was a pretty earth-breaking, groundbreaking sort of a discovery. It got a lot of press all around the world. We had interviews with BBC, with other European news agencies all around the US. And this again is 2024-ish years ago. But yeah, that Adelita story, I think, was one of the neatest stories back in the day. And moreover, I think what it did too is, you have to understand, at the time, back in the early 90s through mid 90s, is Baja California and northwestern Mexico was not really on like the turtle map, so to speak, for turtles. We knew that there's huge extraction, but from a scientific standpoint and from an everyday sea turtle biologist standpoint, Baja was not really a spot that we thought about. Australia, Japan, the southeastern United States, that's where a lot of the bolus of turtle biologists were from. So there are these other sort of outcast areas that were a little bit more forgotten. And Adelita put Mexico, and particularly northwestern Mexico, on the map.
Right. Yeah. That was, in terms of press and sort of, that makes a lot of sense. So talking about satellite tracking and flipper tags, I mean, that's sort of the technology has increased exponentially now with the data you can collect, and how you're actually tracking the animal. I'm curious what the most interesting insights and maybe counterintuitive things you've learned from doing that sort of analysis. And also, I mean, one of your papers was really a cool thing. I remember looking at this early on, as just, as you've mentioned, you can see the movements of the turtle, but you're not seeing the actual what the turtle is seeing. But you can do that if you have mounted cameras. And one of the analyses, you got some connection with the National Geographic Society, and we're able to actually get a visual on what the turtles are seeing. I mean, maybe you could kind of expand upon those things, and what you've learned from tracking, the counterintuitive things from that, and then what it's like to actually see what the turtles are doing in real time.
Mm-hmm. Yeah. So from just the tracking point, some of the neatest discoveries, beside this fact that turtles can sort of rival our whole concept of what long distance migration is like, that they can actually, these animals are swimming tens of thousands of kilometers to get from point A to point B. It's just the reciprocal of that too, though, is that there are some tags that we've put out, and I'm thinking of green turtles in the Galapagos Islands that I've been involved with, hawksbills in the Eastern Pacific and El Salvador particularly, that telemetry can also reveal non-migratory animals. And this is the stuff that's not in the textbooks. We read the paradigms that sea turtles are highly migratory and they're always moving between nesting beaches and foraging areas that are really distant from each other, but it's not always the case. And telemetry is one of those examples that we can, one of those tools that we can use to exemplify that. And the Galapagos case, for example, I think what it did is it shows, and Galapagos has such a high density of turtles, and they're growing at very, very slow rates. This is work that Derek Green did back in the 1980s. And what we realized through this non-migratory behavior is that actually there's sort of, now we see the mechanism why. So density dependent, slow growth because of food, like lack of food. When you're non-migratory and you're in an area that has a bunch of turtles and you just aren't programmed to leave, you are stuck with the resources you have locally. And so that can sort of precipitate out to extremely slow growth rates. And as Derek Green pointed out, these growth rates of 0.1, 0.2 centimeters a year, which is very slow for sea turtles. So that's the I think those are some of the gee whiz things for the telemetry aspect. But Michael, as you point out with the video component, it's kind of like the picture will tell 1,000 words. Well, a video will tell 1,000,000 words. And so in the case of sea turtle research, I was fortunate enough to work with National Geographic in the 90s through the early 2000s, particularly with two colleagues named Kyler Abernathy and Greg Marshall. Kyler is still with Nat Geo and doing a lot of really cool work. But we put cameras on green turtles in the Gulf of California in my former study area, and just learned what the neat thing about a camera is it gives you the turtle eye view to understand the choices that an animal makes when it's presented with options of what habitat or what prey item to engage with. And when you can literally see, and the way the cameras are mounted on turtle, you can actually see the turtle kind of move its head and inspect the sea floor. And you can see the choices they make. And what that does is it helps us get a much better understanding of something we call like foraging selectivity, looking at like what are they selecting out of the available foods. And more recently, with some work that now let me back up and say is that the Critter Cam, which was the Nat Geo system 30 years ago, it was analog. We used high-8 video cassette tapes basically, so it was literally like it was not solid state by any means. They'd fail a lot, and they were big, but we would still get some amazing information. More recently, the video systems that are used are solid state. People make, on their back in their workroom bench, they'll use GoPros and make their own cameras. I think that the sort of the state of the art for off-the-shelf componentry and integrated camera systems that have telemetry and video is something by an organization called CATS, Customized Animal Telemetry Systems, out of Australia. We use CAT systems here in the Eastern Pacific in San Diego Bay. And we found some other really cool behaviors with the video as well. And to me, the coolest thing is the social behavior. And so we, again, let me just rewind for a second. And in our turtle community, in the sea turtle community anyway, the idea of sociality in sea turtles has been poo-pooed by the establishment for a very long time. Even though in the tortoise world, especially since the 1700s and the 1800s, there have been observations of vocalizations, of apparent different sounds, up to 18 different sounds that Gio Cheloni would make. More recent work with Camila Ferrara in Brazil with South American river turtles. They've identified 11 different sounds. There's actually have had some observations that adult females will hear hatchling sounds and it will have them attract to those hatchlings. And actually they believe that South American river turtles, Podocnemus expansa, will use sound to congregate and migrate together. So in the freshwater turtle world and tortoise world, at least, this idea of potential sociality has been around for a very long time. In the turtle world, it just hasn't been there for whatever reason. It just hasn't been picked up as a potential. And perhaps that's because they live underwater and it's a little bit less measurable than at least with some of the terrestrial animals. With our video work, we found in San Diego Bay, for example, that turtles are extremely social. They interact with animals all the time. Intern of ours, Cameron Mullaney, just published a paper in Ecology and Evolution that identified social behaviors and the interaction of turtles, the apparent attraction of turtles to anthropogenic structures for scraping and scratching and cleaning their sides, much like a cleaning station for turtles in areas like Hawaii. And so that video system, to me anyway, has really sort of opened new doors and for me raised new questions about sociality in sea turtles. That's something I'm going to be talking a little bit about. I have a talk coming up next week at the Freshwater Turtle and Tortoise meeting, and I'm going to talk about sea turtle and turtle sociality in general. But I think that's a new frontier that we should really look into. The idea potentially that sea turtles in this case, use some sort of social cues to aggregate and perhaps even migrate together as with river turtles would just be, it'd be earth-breaking. And so we'll see what happens, but it's all because of the video system that has allowed us to ask these new questions.
Well, and that opens up a really interesting question about their navigation. I mean, you think about the natal homing and philopatry associated with turtles. I mean, that could be an explanation for how do they figure out how to get back to where they were born, or to get to foraging grounds with a really high degree of precision. I mean, it could just be that there's a communication element to some of that, right? And compounded with other magnetoreception, that sort of thing. But yeah, that's a really an interesting area. And one of the past guests that we had in the podcast is a student currently, and well, he actually just got his PhD, but he had written a large paper looking at sort of the evolution of sociality in over 50 species of freshwater turtles and tortoises, and vocal communication, vocalizations. And one of the best talks I've ever seen at one of the TSA symposia, and just an incredible analysis. But there was no sea turtle, anything in there, because a lot of this was based on captive animals or things he could get easy access to. So that's a huge area of broad interest, I think. And that's really cool that you observe that.
Can I unpack that a little bit more from the sea turtle side?
Yeah.
So one of my theories for why our community has not picked up on the sociality aspect, I believe, is the scientists that really first kind of raised this possibility was Archie Carr, the sort of grandfather of sea turtle ecology. And back in the 60s, he proposed what was called the social facilitation hypothesis. And what that is that on the foraging grounds, inexperienced females would link up with experienced females and follow those experienced females back to the nesting beaches. Of course, this was at a time when we had no idea about navigation and the magnetism and magnetoreception and things like that. And so he had this idea of social facilitation as the mechanism for how animals would get back to the nesting beach. And starting in the 80s into the 90s, when the genetic techniques started to get refined, is we realized that if it was a social facilitation framework, is that like there may be some randomness to it. So animals from different sort of nesting beach origins would link up, and maybe a female, a newbie, a neophyte female from nesting beach A would follow a female from nesting beach B back to nesting beach B's natal grounds, which would move it from the turtle A from its original spot. What we found, though, from the mitochondrial DNA is that actually animals from nesting aggregations share in mitochondrial DNA, and there's some genetic separation. And so Archie's social facilitation hypothesis was sort of disproven. And I believe that was like that first litmus test of sociality and our concepts of sociality in turtles. And because it was so resoundingly sort of shot down because of very strong empirical genetic evidence, maybe we just kind of abandoned the idea of sociality in turtles altogether, almost sort of this knee-jerk, and just never revisited it. But there's some papers, Johann Chevalier just published a paper not too long ago about sound and turtles, and the fact that sea turtles are able to pick up a lot of sounds, and maybe that aspect is something that can be used in management for bycatch reduction, for example. So yeah, I think sea turtles are social. We don't give them enough credit for it, but hopefully we will in the future.
Right. Yeah. And that's interesting to hear about that. Maybe it's not so much those longer term movements, and maybe who knows, maybe there's some aspect of it that is, but there's so many areas where sociality could be an advantage to an individual. So, well, a group too. I mean, that's kind of a debate, the level of where selection works on social behavior. But yeah, just a really interesting area to look at. And I think maybe that leads us into another area that's kind of interesting, as you mentioned. I mean, the crossover between sea turtles and freshwater turtles. And that's something that there isn't a lot of that. And I think some of that, I'd be curious to hear, but stems from there's differences in sort of the management levels of both groups and the broad habitat differences. I mean, you're dealing with completely different sort of mediums that these animals exist in. And that mandates different levels of management. But you're someone who crosses over between both. And I'm curious if you think there's more, that there should be more crosstalk between both of these areas of turtle science.
Mm-hmm. Yeah, I think that sort of separation is probably just the fact that we are in such a time of increased specialization that we silo ourselves and like really hyper focus on specific taxa or specific themes or specific research questions. If we rewind to 30 years ago, some of the sort of forefathers and mothers of sea turtle research, at least, like the Karen Bjorndals, Colin Limpus, Haitao Shi from the US, Australia, and Japan, respectively. Those are colleagues that have worked on both sides of the aisle for decades. And so there was this trend of scientists looking at multiple turtles of freshwater, tortoise, and sea turtle taxa for a long time. We've lost that today, though. I totally agree. There's very few people that do that. I think there's great opportunities for cross-pollination, both for we just talked about sociality, for example. I think that's one area. I think from a technological standpoint, probably the sea turtle realm has helped advance some of the technologies, whether it's genomic techniques, molecular techniques like isotopes that really started in the sea turtles before freshwater turtles. Those sorts of things, telemetry, battery miniaturization, those are all things that I think the animal platform was sea turtles more than it was freshwater turtles and tortoises. I think both communities have a lot to gain by working more closely together. I think also in a fundraising standpoint, I mean, turtle lovers are turtle lovers. They're not discriminatory between any given taxa. Of course, we all have our favorite species, but we all love turtles. The meme out there, I like turtles. So I think we have a lot to gain from working closely together. I hope that leaders of the TSA and in the sea turtle side of things, like the International Sea Turtle Society, recognize that. I'm not in those leadership roles to promote those things, although I'm a big advocate for looking across the aisle. That's something I referred to the meeting next week. That's the TSA meeting that I'll be giving a keynote at. I'm going to talk about this very theme, actually, of how we can work together to find a greater common good between all turtle taxa.
That's awesome. Yeah, there is that's a great point, just with the general sort of methodologies. I think that sea turtles have broadly gained more attention in a lot of ways, and the development of different methods. I've done some population analyses and that sort of thing with freshwater turtles in the past, but have looked at papers focused on sea turtles. And I mean, any given paper, you open it up. I remember looking through a ton of different freshwater turtle and analyses, and they're kind of basic population models, and then looking at one sea turtle paper that the method section for this stage-based population matrix was 10 pages long, and it had all these novel adaptations of the method. And it's just like I think that it would benefit both fields, to look at how the methods are being used in the sea turtle realm and sort of extrapolating that and applying it, because there is a lot more sort of, I guess, novel approach in some of those areas, just due to maybe more people working on it. So yeah, if we could, I think maybe you mentioned the working with stable isotopes and getting an idea for the diet of sea turtles, but also kind of indirectly other aspects of their ecology. I'm curious what your sort of highlight projects you've worked on as the director of the Stable Isotope Laboratory, what those are, and what maybe technological advancements you're excited for in the realm of stable isotopes in the future.
Great. So to set the table, for those that aren't familiar with stable isotopes, these are differing forms of an atom that have different numbers of neutrons in their nucleus. So what that does is create different atomic weights, and those different atomic weights actually result in animal physiology treating those different isotopes in very different ways. And so from collecting a small quantity of body tissue, whether it's a skin sample or a blood sample or a nail sample or keratin, you can tell something about the biology of that animal. So for example, traditionally, isotopes were used for sort of the first question that was addressed with isotopes was this idea of where does an animal fit on the herbivory to carnivory spectrum. So animals that have higher, for a variety of isotope dynamic reasons that we don't have time to go into today, I can simply say is that carnivorous animals have a very different isotopic value in their tissues than do herbivorous animals. So you can collect a bunch of body tissues from a bunch of turtles within one population, for example, and tell something about the variability in dietary strategy at the individual level. And indeed, we found with isotopes, and this is a resolution that from lavage and fecal sample, we just haven't been able to address because those techniques are more labor intensive, they take a longer period of time, and that results in smaller sample sizes. But with isotopes, it's just I generally use skin for sea turtle work, although we'd use blood for some questions. But you can go out to a field area, collect, capture 30 turtles, collect a skin sample, and then you can do the isotope analysis of skin sample for 30 turtles in a relatively short period of time. Whereas to do the lavage for those 30 animals would be much more labor intensive and probably less, possible to do in like a field setting. And so the traditional techniques for isotopes have been used to address questions about trophic status, trophic ecology. More recently, isotopes have been used to answer a lot of different sorts of questions, like things like animal movement. And so, for example, one of the questions for animal movement is looking at what's the origin of an animal that was captured in my own study area. And so in our world, we have, because of changes in oceanographic and biological and oceanographic processes, we have what are called isoscape maps. So there's these gradients in whether it's nitrogen or carbon or oxygen, hydrogen, and sulfur. Those are the 5 we typically use, but there are gradients in oxygen, hydrogen, and nitrogen particularly, such that if animals that are resident to a given area, if you were to collect their skin sample and you know what the background isotope value is for that area, with this thing called discrimination, which we won't get into right now, but if we were to know what it should be in an area, and we know what it should be for turtles, and you collect a skin sample and run the isotopes, you might encounter some turtles that have radically different values than what you'd expect. And the reason for that is because those animals didn't build their body tissues in your local habitat. They built those body tissues in a different area where the baseline isotope values were different. So in that framework, I believe that one of my favorite projects was a project with leatherback turtles nesting in Indonesia in a place called Jamur Spemeti. It's probably the largest nesting beach for leatherback turtles in the Pacific today. And that's a site that we've done a bit of satellite telemetry. We've got some sense that animals from Indonesia, way across the Western Pacific from where I live in San Diego, California, some animals would leave the nesting beach and go to Japan. Others would go to the high seas of what we call the North Pacific Transition Zone. Yet other animals would swim all the way to the Eastern Pacific to forage. Like, why would a turtle, if it can forage 100 miles away, swim 5,000 miles away to forage? And so these questions about animal movement raise a lot of interesting questions about resource utilization and just the drivers of long distance migration in general. But back to this story about leatherbacks is that it was more of a fishing trip. I just wanted to go collect skin samples from these leatherback turtles. This is in the early days in the 2006, 7-ish timeframe. Collected all these skin samples from nesting leatherbacks in Jamur Spemeti in Indonesia, brought them back to the US, ran them, and I found that, wow, there were some animals that had these nitrogen values in their skin tissue, which were really, really high. And the other half of the animals had these nitrogen values in their tissues that were really, really low. What is going on? Well, what we did then is we sort of tried to sleuth it out and look at the oceanographic processes throughout the Pacific Ocean. And what we realized is that in the Eastern Pacific, so off the coast of California, for example, we have what's called advection of these high nitrogen waters from more southern areas in the Pacific, like off of Mexico and further south, that take this underwater northbound current that brings this high nitrogen water up to the coast of California. And when leatherbacks are foraging on jellies off the coast of California, they're then integrating those high nitrogen values into their own body tissues. But that like high nitrogen signal, it's only present in that one area of the Eastern Pacific off the coast of California. Throughout the remainder of the Pacific Ocean, there's no high nitrogen values anywhere. So we came up with this hypothesis that, wow, this sort of dichotomy, this bimodality of high and low nitrogen, maybe those high nitrogen animals are coming from California. They forage in California, and the low nitrogen animals were foraging everywhere else. And so we came up with an experimental design to go out and randomly place satellite tags and collect skin tissue samples from roughly 30, 25, I can't remember the sample size today, but about 25 different individuals, and let them go. And sure enough, the animals that came back that had the high nitrogen signals from those skin collections that we did right when we were putting the tags on were the very animals that swam to the Eastern Pacific, and the turtles that had the low nitrogen values were the animals that stayed in the Western Pacific. So that was came out in 2012 in PLOS, and that was the first paper that really showed the value of what we call the isotope tracking technique for sea turtles. And since then, it's been used in a number of other examples. If we have time, I can share one other example, which is pretty cool. But that sort of shows how a molecular technique like stable isotope analysis can be used to answer questions that are hugely germane to management. And so maybe you can't say, okay, this turtle went to microsite A, B, or C, because that's the specificity that a satellite tag will give you. You can see right where it went, but at least you know from isotopes that it went to that area. And if we can start to decipher out like this dichotomy of how many turtles go to the Eastern Pacific versus the Western Pacific, then we can start to ask questions about susceptibility to some of those spatially explicit threats like fisheries bycatch. And so in the early days of isotope analysis, and I work at a US federal agency that's focused on management. And so the questions that I address have to be management oriented. And since day one, I've been trying to, get to the top of the highest mountain and scream that isotopes can be used to address management questions. And indeed, that is one really great example, the isotope tracking technique that is indeed something that can provide insights for management.
Right. And it's really interesting. It's kind of a tool to let you develop new research questions and kind of refine and get at different aspects of ecology. Yep. I think that maybe segues us a little bit into kind of the area of conservation and management itself and what that's like to actually implement. And so you've been part of the IUCN and ESA status assessments for the green turtle, and at least an author on the loggerhead analysis for ESA. And I'm curious kind of what were the most surprising things you learned in the process of compiling that information?
I have been, well, I haven't lately, but 20 years ago, I was one of the biggest doubters of the IUCN Red List technique. The IUCN Red List in general, to use a single label to identify the conservation status of a globally distributed species that has multiple individual demographic units and nesting beaches. I think it's using a sledgehammer instead of a scalpel. And for management, you kind of need to use the scalpel in a lot of cases. And so I was sort of one of these proponents that said we have to focus on other ways to identify conservation status in terms of spatial resolution. It can't be just at the global level. And thankfully, not through my griping, but I think in general in the sea turtle community, both with the U.S. framework, the Endangered Species Act, and with the IUCN Red List framework, there have been allowances now to identify this conservation status at subglobal levels. For the Endangered Species Act, we call these subglobal units distinct population segments. And then sort of the analog for the IUCN Red List framework, we use the term called regional management units. So DPS's distinct population segments and RMU's regional management units. And both of those, what those do is we're able to then slice the global pie into separate regions, recognizing through whether it's telemetry or genetics or flipper tagging or other empirical data, like where are the separations between these population, these component populations. And so for the IUCN Red List and the ESA work for green turtles, we were able to identify, and this was not surprising, but something I think it was helped us illustrate the need to change how we do business. But back in the early days with the Red List, at least in 2000, so 24 years ago, we identified that there's vastly different population trajectories, population trends, depending on where you are. Some places, green turtles would be extremely endangered, like in the Eastern Pacific back in the day, and other places, green turtles would be doing very well, like in the Indian Ocean back in the day. And so to be able to separate out those different conservation statuses, I think is a really valuable thing when it comes to management, because we want to have the best available science, not for just the general, prototypical sea turtle, but we want it for the turtles that live in our backyard. And so to me, those status assessments really open my eyes more and more to the need and the recognition that turtles are doing from the same species might be doing very different things conservation wise in different regions worldwide.
Yeah, the sea turtle situation, the biogeography does call in. I mean, it brings a big question up in terms of scale of conservation, right? The RMU concept, and yeah, with the ocean, it's one contiguous thing for the most part, but also the way the animals are distributed isn't. So you have to think about, yeah, how do you most effectively manage and preserve the phylogenetic diversity and the unique sort of adaptation of it. That sort of thing all comes into question, and defining that is sort of challenging. Um, I'm kind of curious, just overall, talking about that scale. I mean, you have experience working in a lot of specific places with sea turtles, like the Gulf of California, but also at a global scale. And maybe we could just start out conservation wise. Maybe in the Gulf of California, that's an area you've got a lot of long term experience. I'm curious how the conservation status of all of the sea turtles within that region has changed over time. And then maybe we could extrapolate that out to kind of the global scale,
Mhm.
and take that however you want to, sort of whatever direction.
Of course. Yeah, well, we can start with green turtles. So, um, recall at the beginning of our podcast, I talked about Los Angeles Bay in the early 1990s of how there were so few turtles. This was 3 years after the President Salinas de Gortari declared a moratorium on turtles, but that was a decade too late. Um, for us, my PhD and subsequent postdoc work in Los Angeles Bay, it ran from like field collections ran from 1994 to 2004. And of all those years, and I was fortunate enough to have the support of an organization called Earthwatch, which some of our viewers might know. It's this sort of eco volunteer thing where you pay to go to a project and help them do work. In some places, it's kind of busy work. The scientists want the money, but not the volunteers that they have to deal with, the babysitting. For me, it was totally kind of opposite. Actually, we needed the people power. We needed the money, but we needed the people power because we were running these things called entanglement nets. It's the style of net that's used to capture a turtle. But we were running 6 days a week, 24 hours a day, 8 100-meter nets at a time. So 6 days a week, 800 meters in net for 24 hours a day. And we would do that for 3 months straight. So do the math on that. I'm not good enough at math to get that out right at the moment, but the most that we captured in any one year was in 2001. And it was a long year. It was about 3.5 month season. And we caught 50 turtles, green turtles. And that was a good year for us. We were completely stoked. Fast forward to today, and we can get into the potential reasons why it's the case, but green turtles in the Eastern Pacific, much like around the world, have been making this almost miraculous recovery. Florida is another example. Green turtles are through the roof. In the Eastern Pacific, green turtles have rebounded in such a powerful way that we see them in nooks and crannies all over the place, even up here in California, in spots that we would never have expected to see turtles. Back to Los Angeles Bay, what that results in, to give you a sense, is that a couple of years ago, my colleague Erica Santa Cruz, who now runs the sea turtle work in Los Angeles Bay, she was doing work. She put one 100-meter net in the water, and in less than an hour, she caught 80 turtles, and had to pull the net out. So I don't know, it was 45 minutes, but it was under 1 hour. She caught 80 turtles. In the eighth of amount of nets that we used, that it took 3 months to catch 50 turtles. So we're talking at least 3 orders of magnitude greater numbers of green turtles in LA Bay, in the Gulf of California, probably, maybe even more. And so we see that sort of same rebound with green turtles in many places. And is this just a fact of changing climates? That might have some influence to it. I tend to think it has more to do with the fact that fewer people are eating turtles today. There's more conservation attention, less egg harvest and egg consumption is happening. And back to my point about resilience, if you leave them alone, they can do really, really well. And so green turtles, I think, are the poster child of how effective conservation has led to incredible population recoveries. Don't get me started on how that relates to the fact that we still call them endangered species. That's for another podcast. But nonetheless, it's a really promising aspect. Olive ridleys are another species that we're seeing incredible rebounds. Loggerheads in Florida they go up and down, but numbers are very high. The species that's most problematic when we talk about conservation status that just hasn't made the rebounds that we would have hoped are leatherback sea turtles. And leatherback turtles, for the longest time, we've known that in the Pacific Ocean that leatherbacks have declined at a very unsustainable rate. And to give an example, a place called Las Balas National Park, Playa Grande in the state of Guanacaste in Costa Rica, was one of the biggest strongholds of leatherback turtles in the Eastern Pacific for nesting. The other sites were in Mexico and to a lesser extent in Nicaragua. But in Costa Rica, don't quote me, but the numbers, let's say from the 1980s, was roughly like close to like 2,000 nests a season, probably more than that. They had a huge, well, it was called Las Balas, that means leatherback in Spanish, Las Balas National Park. So it was a national park named after the species because so many animals came to the shores. In the last few years, it's like less than 10 individuals come to nest. And I think last year it was like 4 or 5 individuals. Again, don't quote me, but it's less than 10, single digit number of animals. The population has completely crashed. Why? Well, it was egg harvest early in the day, but probably more importantly, more detrimentally these days, it's the incidental bycatch, the accidental capture of leatherback turtles in artisanal and industrial long lines and drift nets. So we thought with leatherbacks, okay, we know they're tanking in the Pacific, although there's a little bit of a stronghold in Indonesia where there's about like 2,000 nests a year right now. So it's kind of still trebling along. But in the southeastern US, the western North Atlantic, we thought that the population was doing quite well. Well, all the western North Atlantic leatherback populations have also crashed. And so we are seeing leatherback turtles that, just at fractions of their numbers. My colleagues that for years worked in St. Croix in the US Virgin Islands, for example, they used to have huge volunteer programs for leatherback turtles. Leatherbacks barely even come to that beach anymore. It's like also less than 10 animals. So we're seeing really alarming patterns for leatherback nesting abundance, negative trends. Those causes are probably related to human take, but there might be a habitat component, a climate change component to leatherbacks for a variety of reasons that we don't have time to talk about today. But I mentioned leatherbacks just to point out, to juxtapose that, you have this hard shell species like a green turtle that's doing really, really well. And then you have the leatherback, which is doing very poorly. And so in the turtle realm, it's the sort of two ends of the spectrum. When you talk about leatherbacks and the low numbers, yes, this idea of endangered status definitely comes into vogue because they truly are endangered with extinction. And indeed, like the Costa Rica population might just blink out altogether in the foreseeable future. So that gives a little bit of a taste of some of the trends. And there's things in between. Loggerheads are doing different things around the world. Kemp's Ridleys go up and then the oil spill happens and they go back down, but they're kind of copacetic. But yeah, green turtles are doing great. Leatherbacks are doing very poorly. And that's sort of a snapshot.
Right. It kind of varies a good deal according to species. And the leatherback situation is, that's something that's really interesting. We've talked to a few other folks, Dr. Jeanette Weineken, a bit about some of the work she's been doing on sort of the hatchling leatherback failure that's higher than most other sea turtles and what might be the selenium interactions that may be leading to that sort of thing, and all these different little nuanced factors in their biology. And it seems like maybe it's just a bunch of different angles they're getting hit from, and it maybe isn't one particular thing. It's just they're more vulnerable, which is, it's really unfortunate. But so in terms of the future, I mean, you've served in a lot of different leadership capacities in the sea turtle world over the course of your career, and you still do. I'm curious, what are the trends in sea turtle science and conservation that make you the most worried, and what are those that make you the most optimistic? And how do we tackle the problems that are facing sea turtles at a global scale?
Mm-hmm. I think the thing that makes me most worried is this sort of unabated global bycatch in the high seas that happens by perpetrating nations that do not engage in international conservation dialogue. I think we can all identify probably who those nations are. I don't need to get into that right now, but safe to say that there are nations that send industrial boats around the world to put literally millions of hooks in the water Some of those fishers don't see shore for years. They stay out on these motherships and just catch and catch and catch and catch with no interest in trying to be sustainable or limit incidental or accidental capture of non-target species like turtles or whales or dolphins. And so to me, at least in the conservation realm, I believe that the industrial high seas fisheries are the biggest nut to crack. We have some players like Spain that are starting to come to the table and dialogue with us right now. So I know I want to applaud Spain for that, but there's other nations that really have shown no interest in engaging in the national fora for international fora for talking about bycatch reduction. But to me, focusing on the positive, I'm a very optimistic person. I think it's just the cadre of committed people, whether it's nesting beach volunteers, people working for free because they love turtles. And that love for turtles, it's being expressed with sea turtle examples, but this is another case where I believe that some of the volunteers and that happens in the sea turtle realm could be transferred to the freshwater turtle and tortoise realm and really have some positive influence. But I'm just so sort of, I feel such a sense of like enrichment and optimism just based on our community and how much dialogue is out there. It used to be this big sort of global network. The global networks, sometimes they get too big and there's not enough opportunity for the small person to have a voice at the table. But fortunately, there's an increasing number of smaller regional conservation networks, regional conservation groups, where at least scientists can feel like more of a sense of ownership and they can coordinate and collaborate with people that live in neighboring nations or neighboring communities to solve common problems. And so that idea of just the huge commitment on the part of the turtle community to solve these problems is the thing that lets me sleep at night.
That's awesome. I think we'll finish up with a few questions just related to, we focus a lot about the research and kind of the experience doing that, but there's some things that don't make it into scientific papers that are interesting to touch upon. I'm curious for you personally, out of all your experiences with sea turtles, what's the most memorable and why?
Well, the most memorable is the comb-cut green turtle one, without a doubt.
Yeah.
I told that story, but the second one is the one that for everything I've done, the most exciting project that I have been part of was the discovery, the Western science discovery of major hawksbill nesting beaches in the Eastern Pacific. We had for many years seen juvenile hawksbills swimming in waters from Mexico throughout Central America, down into Ecuador, even out to Galapagos Islands. Thought there was just a smattering of solitary nesting females that must have been the source of these hatchlings, a source of these turtles that we saw in the water, pardon me. And then in 2008, at a sea turtle symposium that my colleague Jay Nichols was the host for, the president of the society at the time, we had a colleague who, his name was Michael Lyles, who now is a NOAA colleague of mine, but he came to the meeting and he was bringing a data set. He was going to do a poster, and the poster was on hatching success of hawksbill turtles in El Salvador. And he was never been to a meeting before, no turtle meeting, super nice guy, a little bit quiet, a little bit timid. And he came in, he's like apologetic, like, I have these few data. And Jay and I and my friend Brian Wallace, and I was like, we almost fell out of our chairs. Like, what? There's a hawksbill nesting beach where you have information for, like, to the extent that you've studied hatching success? It absolutely blew us away. And that was a meeting that occurred in January in 2008 at NOAA at the time. I'm like, oh my, this is a big deal, because hawksbill turtles we thought were on the brink of extinction in the Eastern Pacific. So I found funds. We assembled a team of folks to go down and have a hawksbill working group meeting for sea turtle scientists from throughout the Eastern Pacific, a lot of different nations. And we started to put together, like, you have a little bit of information and you have a little bit of information. There's a couple of turtles here and a couple of turtles there. And we started to formulate this sort of profile, to characterize hawksbills in the Eastern Pacific. This is all in the backdrop of, with Mike's work, we discovered a major hawksbill nesting beach in El Salvador. And then a short time later, we found another major nesting beach in Nicaragua. So right side by side. Why didn't we see them before? Why were they undiscovered? The reason is because they do not nest on sandy nesting beaches. They nest inside of estuaries in the mud under mangroves. How the embryonic biology works that these animals can survive in high humidity mud, low oxygen levels, that's for another day. But we realized we were looking in the wrong places. And so through that work with Mike, and again, that came from local ecological knowledge. Mike worked with because egg harvest is legal in El Salvador, but the conservation biologists were providing funds for egg poachers, egg collectors to go out and bring the eggs in for the scientists. And little by little, we amounted all this information. And today, and it's actually started in 2008, there is a conservation network called ICAPO, in English, it's the Eastern Pacific Hawksbill Initiative, that now hawksbills, we've identified 3 major nesting beaches, a couple of secondary nesting beaches, some major foraging hotspots that we never knew about before. Hawksbills are no longer on the brink of extinction like we thought they were. And it's a very exciting discovery that I still remember those nights on the beach, seeing the first ever hawksbills to Western science crawl out of the water to nest. It was amazing. To this day, I mean, it's one of my, along with that green turtle story with the comcock, it's my most cherished, most exciting memory. And also because it happened with a bunch of great friends.
That's awesome. And so something is sort of the second to last thing here, right? The sea turtle work, I think, brings out a lot of philosophical sort of takeaways. That's something interesting to me, is like the broader context of, why sea turtles and why devote sort of life to conservation, that sort of thing. I'm curious, what are some of in doing field work and analyzing data, those sorts of things, what are the broader kind of philosophical takeaways? What have you been kind of forced to reflect upon in doing a lot of this work?
For the longest time, I have been sort of a traditional biologist focusing on collection of empirical data from turtles. Yes, I engaged with turtle fishers and had those fishers teach me everything I knew, working closely with them. But what I've found more recently through some other work that I've been engaged with, fisheries bycatch work, is that at the end of the day, like, the amount of data we have is almost secondary. It's really, how do we enlist the maximum number of people to kind of help conserve the species? And so I'm absolutely convinced that social science is more important today than biological science. And I think social science is the realm, that's the frontier. That's whether it's communication strategies, on broad terms with creative techniques like conservation radio, where you can broadcast to fishers on sea that are bored and all they have is their radio, and you can give conservation success stories or conservation stories, or whether it's working with enforcement agents and building their capacity to better enforce trade of freshwater turtles and tortoises in the pet trade, for example. I believe it all comes down to the social component. And that's really where a lot of my work is starting to focus, is on the social aspect. And I think human behavior is especially important. And, this integrative need to go beyond biology and to bring people into the mix and social science into the mix, that's the number one thing that I think about more and more all the time.
That's awesome. Right. Spreading the message and recruiting people in conservation is, I think, a really important thing. You can't change how trajectories are going if you don't have buy-in from the majority, I think, generally. So it's really interesting, and that's cool to hear that that's sort of the future. And then the last thing I'm curious, and just as a younger person in conservation and everyone that sort of works in this podcast, what's one piece or a few pieces of advice you'd have for someone who's looking to make turtle work part of a career or a career?
Well, I think the first thing is that we have that pressure from our parents that, oh gosh, there's no jobs. You can't find a job in that realm. And I totally disagree with that. I believe that if you're the best at what you do, and you've built your skills, that you're going to find that job. You're going to find a way to make a living. And for what we do, we're not in it for the money, but the reality is you need to pay the bills. So, I think it's stay the course, make sure that you're working your butt off. That's one thing. The other thing to me, like, and what, Michael, to your question, I'm responding with the things that I identified that helped me get to where I am today. So that's the thing. And the another component, you never take no for an answer because we've all heard no. But it's relationships. We are in a relationship business. This kind of goes back to my answer about the social component being so important in conservation. But for us as well, as we're building our careers, as we're trying to create new opportunities for ourselves, the only way we create those opportunities is through human connections. And so you never know what some new acquaintance that you meet it's going to lead to later on. And so I've always thought that, when we're meeting people, whether it's at a scientific meeting or on the street corner or in a class or, it's a professor or something like that, I try and be authentic and genuine and like give them my attention and be earnest in my engagement, because it's like putting a little paper boat on the stream and like they all go down the stream. You never know what's going to come back to you. And I know that because for me, with my current job at NOAA, that was one of those little paper boats that I sent down the stream. It was my friend Ana Barragan from Mexico that I had become friends with. And she knew closely Peter Dutton, who's the guy that hired me at NOAA. Peter didn't know me, but he knew Ana. And he said, hey, I'm looking for somebody for this position. And Ana knew me. So it's like personal relationships are just so important. And then my final take home is that find a mentor or find 4 mentors and recognize that there's mentors and recognize that you can learn a lot from them and engage with them, talk to them, have a coffee with them, keep in touch, like reach out occasionally, because mentorship is just such an important thing. It's like, it gives us a little bit of a head start by talking to somebody and finding out how they got from point A to point B in their own careers. It can help us make a roadmap for how we want to get to what our ultimate destination is as well.
That's awesome. I think I'll take that advice to heart for sure. And we've just wanted to sort of close out this. We're currently at the Turtle Survival Alliance meeting, and we wanted to close out this interview with sort of a dedication to Dr. Jay Nichols, who is just an incredible mind in sea turtle conservation and had a knack for the philosophy of sort of nature and the ocean. It's super unique and interesting. And he was going to be a guest on the podcast, and unfortunately, a few weeks before, passed away. And so we're here with Dr. Seminoff to talk about his legacy a bit and dedicate this episode. But I guess just to sort of segue into it, what are some of the fond memories you have, and what are some of the adventures? It seems like he was a really adventurous guy.
Yeah, thanks, Michael. I just want to say that, first of all, that we all have gotten to where we are from having close friends and colleagues and shoulders to cry on and minds to throw ideas back and forth with. And for me, for the last 38 years or so, Jay Nichols was that person for me. And, we met in 1993 together. We were both born the same year. I'm 56. I was born in 67. Jay's about 4 months older than I am. And we just became sort of brothers really early on. And so, at this point, to talk about Jay, I was super sad. He passed away in early June, and now I just think about the memories and the positive things, and like I have just whereas my heart was really hurting before, now it's starting to be more like filled with joy and remembering like all the great things that we did together. And so to give an example of the sorts of things is, so Jay and I both, Michael, I can't remember, it's a week ago in my shorter midterm memories shot, but I'll give you a quick example of this. Is that when, and this is sort of my origin story. And did we talk about the day on the beach drinking tequila in Mexico? Okay.
Yes, a bit.
So I'll elaborate on that a little bit more. Is that Jay and I both, when he first moved to Tucson, Arizona in 1993, I had lived there for a number of years already. I did my undergraduate degree at University of Arizona, and we were both starting our masters. And we went to Mexico together to hang out, learned each other just briefly, as you've heard already, everybody, but we worked in Tortuguero together in Costa Rica. We planned this great trip to travel around to all the nesting beaches in Mexico, which was a preposterous idea at the time. Nobody had done it, let alone a gringo. Now, mind you, this was pre-narco days. There's not the same security issues as there are today. But at the time, Jay and I spent months and months to kind of come up with a plan to do this trip. And, little did I know is that here, 30 years later, it's still probably the biggest adventure of my life. And so things of we're driving my 1975 Toyota Land Cruiser. Someday there's going to be a documentary called The Land Cruiser Diaries that is made from this, and we have a lot of old video, which will be great to kind of get into the mainstream. But our adventures spanned from having the rear axle fall off of that vehicle in the middle of the Yucatan Peninsula to literally getting shipwrecked on Cozumel Island, and I'll circle back to that one just a moment, to being held at gunpoint in Chiapas, Mexico, to sitting for 3 days on a dock waiting for a ferry to leave from Mazatlan to La Paz, Mexico. And so those are just a few examples, I think, of just these sort of really formative times, memories that have just totally galvanized and become part of my own sort of formulation story, my origin story. But I will talk briefly about the story in Cozumel. Just last Friday, it's Sunday today, 2 days ago, Brad Nahill, who's been a guest on your podcast, held a remembrance for Jay, and it's the second one he did, and I was able to attend. I ducked out of some of the talks here to get in that meeting, and I feel good today. That other day, I have to say, there's some tears. I've had a lot of tears over the last couple of months, cry out loud moments. But again, I'm filled with more joy than I am sadness at these days. But we wanted to get out. This is August 1993. The important thing to mention too is we brought one of our pets, and that was my dog Rooster, which is a 120-pound red Doberman, unneutered, unclipped ears or tail. He was a big he wasn't ferocious, but anybody that saw him would be afraid of him. So we're on this boat going out to the island. We wake up in the morning to get on the boat. All right, let's go. We're heading out to Cozumel, and in about 45 minutes later, and this is about a 140 to 180 foot long boat. It rocks back and forth and comes to a screeching halt, and we look out the both windows, and the barrier reef of the Yucatan Peninsula is on both sides of us. The boat apparently had lost power or lost control and launched itself, drove up onto the beach. And so we're sitting there, and over the course of a day, various tugboats came out to try and pull us off with no success. And then finally a different smaller tug came out, and they were going to evacuate us from the boat. And now we had left from mainland Yucatan, and we had just gone south maybe 20 kilometers and hit the barrier reef on Yucatan Peninsula. But they evacuated us out to Cozumel Island. And so we had nothing but the clothes on our back and our dog to get out to Cozumel. And we spent a week out there with no food, no money, no anything. We were left high and dry. We got some almost worthless chits, basically food stamps from the shipping company. But that's one of those times that for Jay, and so many other times, he was unflappable and just never got stressed, was the most friendly person in the world all the time. And after that week, we finally got evacuated back to the mainland. And through that incredibly stressful moment, like never a cross word. We did never, unfortunately, get to see a nesting beach out at Cozumel, but we had plenty of adventures and met lots of really interesting people and very just gracious and generous people. And that sort of theme of unflappability with Jay and his easygoing demeanor is something that I carry with me today. And he's not here in body, but he sure is here in spirit. And he's somebody that I just love so much. Jay and I had an opportunity to see each other last November, and so we keep in touch very regularly, or did. But in November, the two of us celebrated the 25th anniversary of an NGO called Grupo Tortuguero de las Californias, which is an NGO that we both started back in 1992, 1993, if you will. And so we just, we had a great time together. And you never, with your closest friends and colleagues, you never think it's going to end. You don't know the finiteness to it, right. Until it's gone. And so for me, I think the take home is to cherish every moment, to love our brothers and sisters and tell them that we do, and work hard but play hard together. And so for Jay, I'm just, my heart is sad, but it's also filled with joy that I know I'm going to carry him with me for the rest of my life.
Thank you.
Absolutely. It was a tough one. It and our whole sea turtle community. Much like in what I've seen in my much less time interacting with the tortoise and freshwater turtle community, but I see such a sense of like brother and sisterhood, and like everybody gets along, and support and mutual support, unequivocal support, unconditional support. The sea turtle community has the same thing. We might be a little bit larger, but we have very much the same thing and a lot of support. There's just such an intersection in mentality between these two communities that I can't wait to see a day when we all work closely together. But Jay is one of those people that his message and his vibe, like his positivity, would have rung true on the tortoise and freshwater turtle side of the house as well. So
that's awesome.
Yeah.
Well, thank you for being here and helping to bridge that gap.
Absolutely. Absolutely.
That's it's really cool to see. It's one thing to say that those gaps are going to merge, but it's another thing to actually do it. Mm-hmm. And so that's really cool to us. And thank you for your work, and we'll honor Jay's legacy through this podcast.
Thank you, Michael. And yeah, long live the memory of Jay Nichols. He was a brother.